Okay, everybody, let's take a few minutes and actually work through the process of DNA replication. This is very similar, regardless of if we're talking about a eukaryote or a prokaryote. The actual logistics of replication are the same, but please review in your reading and in the module what some other key differences about replication in prokaryotes and eukaryotes are. Okay, so it makes sense to start at the beginning. So let's do that. DNA replication begins at what we call the origin of replication. You can see that here, represented by the phrase origin of replication. And as the replication begins, it creates this bubble. Because remember, what's happening here is that the hydrogen bonds between the nitrogen bases are breaking apart. So as a result, you get what's called a replication bubble. And the replication bubble, again, is this whole area that's being replicated. Replication is occurring in both directions, moving away. Something else to remember is that DNA replication is what we call semi-conservative. And what that means is that you have a DNA strand here represented in blue, and you have new DNA strands being replicated down here in green that retains the original structure of the DNA that's being replicated. Please review your book and other sources to determine who actually discovered the semi-conservative nature of DNA replication. It's in the book, and you can easily find it. It's two scientists, I'll give you that much. So make sure you look into that on your own time. So again, DNA replication is semi-conservative, meaning that you start with the DNA template and you create two new molecules, each of which having half of the original DNA to build the new double-stranded DNA molecule. And you can see that in both this double helix form here on the left, and then on the right where you have the ladder form. Something else to note here is the orientation of the DNA strands. They're what we call anti-parallel. And you can see here that you have a 5' end and a 3' end. So a 5' end of one part of DNA faces the 3' end of another, with its specific base pairing here in the middle. And then the 3' end and the 5' end of the bottom part of it also faces each other. But notice the molecule, 5' end of DNA is here, it ends with a 3', and alternately the other side bottoms out with a 5' and at the top has a 3'. So what anti-parallel means is that these two different parts of the molecule face the opposite side. So what does anti-parallel mean? I keep using that word. Let's look at that a little bit closer. Anti-parallel nature of DNA is related to the nucleotide structure. Go back and review what a nucleotide is, but here's just a short picture, which should clearly look familiar to you by now. But you'll notice that we have this phosphate group here, we have a nitrogenous base, and then we have the pentose sugar. Pentose, again, meaning there are 5 carbons. And that's what these red letters mean. 1', 2', 3', 4', 5'. 5' and 3' mean the number of the carbon that's exposed. So if we look at a small piece of DNA, take note of several things. First of all, adenine and thymine are paired together with a hydrogen bond here. Guanine and cytosine are paired by hydrogen bonds in the middle. Remember from earlier in the module where I asked you to look at how the backbone structure of DNA is formed. You can see that we have a 5' end and a 3' end. On its anti-parallel side, we have a 3' end and a 5' end, and they're facing opposite sides to one another. The 3' end is the side that has an O or an OH on it. You can see that here. The 5' end generally has a phosphate group on it. So the 3' end of one molecule is going to bond to the 5' end of another nucleotide adjacent to it. You can see that here in the bottom. We have this guanine right next to a thymine. You'll see that the phosphate group of the guanine is bound to the 3' OH group of the thymine. So the 5' carbon has the phosphate group, the 3' carbon has an oxygen that's ready to bond with something. And in this case, the phosphate group bonds. We have a hydration reaction or condensation reaction, and as a result you release water. If you are confused by that term, you should go back to the beginning of the semester where you talked about building macromolecules. Okay, so here's this picture that I just showed you before. You've got DNA replication occurring in both directions. I'm only going to focus on one half of this replication bubble, which I'm going to call the replication fork. So as we proceed through the next couple of slides, you need to take note of what exactly is going on. I've used a lot of color coding to help you walk through this process. But again, notice that 5' and 3' are again opposite to one another in every different scenario here. Let's look at it a different way. Again, this is the view of the replication fork. The replication fork has the top side being replicated and the bottom side being replicated. The major components of replication of DNA are here in this key at the top. Enzymes are written in blue and will be color-coded in blue, I should say. And relevant terminology is going to be color-coded in green. So first of all, let's look at the terminology. Template DNA is the original DNA strand that's being replicated. The replication fork is being unzipped and replicated in 2 different directions. And we have other terms that are new that you're going to need to know. The first of which is called the leading strand. Leading strand is the strand that is continuously replicated without stop. The lagging strand is the strand that's being replicated in the opposite direction, but because of how it's moving, it has to take breaks. And we'll talk about what that means in a minute. So now let's look at the enzymes. There are 2 major enzymes that you're going to be required to know about. The first is helicase. Remember, enzymes are proteins, and anytime you see a word ending in ASE, it means it's an enzyme. So helicase is named that way. You can look at how an enzyme functions based solely on how it's worded and spelled. So helicase, because it ends in ASE, is an enzyme that has something to do with the helix. And in this case, helicase is an enzyme that unzips the DNA double helix. And how it does this is it breaks the hydrogen bonds. Remember, hydrogen bonds are weak. Hydrogen bonds are temporary. So hydrogen bonds can be broken easily, and helicase is the way it happens. DNA is unzipped using helicase. DNA polymerase is on the top and on the bottom. DNA polymerase is an enzyme, ASE, that makes a polymerase of DNA, DNA polymerase. DNA polymerase is an enzyme that makes more DNA. It has a very specific job. Enzymes have one job. This is key to how replication works, and you must burn it in your skull. DNA polymerases only work in the 5' to 3' direction. What this means is that, remember, you have a molecule that is anti-parallel, and you have unzipped it now. So you have a template strand of DNA, which is coded in blue, with these green molecules being new DNA. When DNA polymerase lands on the leading strand, it is synthesizing in the correct direction, 5' to 3'. So you can see this DNA polymerase molecule on the leading strand adds a 5' and builds it towards the 3' direction. Because it's moving the right way, it doesn't have to take a break. It keeps working. The lagging strand, as I mentioned earlier, has to take a break. And what this means is that as DNA unzips, you're exposing more and more nucleotides to be replicated. So in this case, DNA polymerase can only land in the appropriate spot where it can lay down DNA in a 5' to 3' direction. As the replication fork continues to unzip, you expose more DNA, so you have to bring in another DNA polymerase to get the job done. So the lagging strand is called that because DNA polymerase can only work in a 5' to 3' direction, and as the replication fork opens up, you have to bring in more enzymes to do the job. Here's another way to look at it and to remind you again how macromolecules are built. You have DNA polymerase, again, which works only in the 5' to 3' direction, as indicated by this giant blue arrow. It works in the 5' to 3' direction because the DNA polymerase, when it lands here, so right now it has an A. Let's look at this A here by the cursor. This A is ready to be bound to a T. So you can see another nucleotide with a T coming in. This nucleotide has an OH group on the 3' carbon and a 5' carbon with a phosphate group. A 3' end exposed here is going to bond with this 5' phosphate group. And what happens is you have too many phosphate molecules. So the phosphates disappear, and also you lose water because you're building a larger molecule in a dehydration synthesis reaction. So what happens is the phosphates leave, water leaves, and then you have a newly bound molecule. 5' is here, 3' OH is exposed here. So when this 3' OH is exposed, DNA polymerase recognizes it and adds a new nucleotide. So with that, you have to build more DNA. But the problem remains is that the lagging strand is missing chunks. The leading strand, again, continuous 5' to 3', always building a molecule. The lagging strand has chunks of DNA because you have many different DNA polymerases working. As a result, we have a new enzyme here called DNA ligase. Ligase pieces together bits of DNA on the lagging strand. And these bits of DNA have a very specific name called Okazaki fragments. Okazaki fragments are only on the lagging strand and are only bound together using DNA ligase to make complete copies. So here's a different representation of everything I've just told you about. You've got DNA replication, you have the template strand, you have DNA polymerase. Up here on the top is the leading strand because it's working in the proper 5' to 3' direction. DNA polymerases here have to work backwards, so you get these Okazaki fragments because helicase is unzipping in the other direction. DNA ligase comes in, binds Okazaki fragments. As a result, now you have two growing copies of the same molecule of DNA. So as I said, the process of replication in prokaryotes and eukaryotes is extremely similar. There's a few very minor differences. First of all, prokaryotes end replication basically when their circular DNA runs out. So here's the origin of replication here. You have one origin of replication. So this circular genome of prokaryotes starts creating another loop, and this continues and continues and continues. And remember that the replication bubble is getting wider as more replication is happening. So then at the termination of replication, you get two circular genomes that pop apart. So the key differences here is that prokaryote replication of DNA occurs in the cytoplasm and has one origin of replication. On the other hand, eukaryotic replication occurs in the nucleus of eukaryotes only. And because the genome is much bigger and linear, remember eukaryotic genomes are linear, there are multiple origins of replication. So the only way that replication will end in a eukaryote is when all of these different origins of replications bump into each other. So you can see this origin of replication building here, 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 and here, and then they start spreading and opening up even more, resulting in two different linear strands of double helical DNA. Feel free to look back and forth in your book, look at the notes, look at some of the module stuff, or rewatch this as much as you want. We can cover this in some of our Friday sessions or on virtual office hours, but please make sure you look through this, take notes, and draw this process out on your own. That's really going to help you out in terms of understanding how replication works.